Hey everybody, my name is Justin Momo. I am a professional pharmacist and founder of Pharma Friend Justin. My motive is to make people more informative and spread awareness about nucleic acid or RNA type COVID-19 vaccine. What are the COVID-19 vaccine types? There are four categories of vaccine in clinical trials. The four main types of COVID-19 vaccines are whole virus, viral vector, protein subunit, and nucleic acid RNA and DNA. Some of them try to smuggle the antigen into the body, others use the body's own cell to make the viral antigen. Nucleic acid or RNA type COVID-19 vaccine. Nucleic acid vaccine uses genetic material, either RNA or DNA, to provide the cells with the instruction to make the antigen. In the case of COVID-19, this is usually the viral spike protein. Once the genetic material gets into human cells, it uses our cell protein factories to make the antigen that will trigger an immune response. The advantage of such a vaccine are that they are easy to make and cheap. Since the antigen is produced inside our own cells and in large quantity, the immune cell should be strong. A downside, however, is that so far no DNA or RNA vaccine have been licensed for human use, which may cause more hurdles with regulatory approval. In additionally, RNA vaccines need to be kept at ultra-cold temperature -70 degrees Celsius or lower, which could prove challenging for countries that don't have specialized cold storage equipment, particularly low and middle income countries. What are the nucleic acid vaccines and how would they be turned against COVID-19? All vaccines work by exposing the body to molecules from the target pathogen to trigger an immune response, but the method of exposure varies. Here how DNA and RNA vaccines work. Nucleic acid vaccines use genetic material from a disease-causing virus or bacteria, a pathogen, to stimulate an immune response against it. Depending on the vaccine, the genetic material would be DNA or RNA. In both cases, it provides the instruction for making a specific protein from the pathogen which the immune system will recognize as a foreign, an antigen. Once inserted into the host cell, this genetic material is read by all cell-owned protein-making machineries and used to manufacture antigen which then trigger an immune response. This is a relatively new technology, so although DNA and RNA vaccines are being developed against various diseases including HIV, Zika virus and COVID-19, so far none of them have yet been approved for human use. Several DNA vaccines are licensed for animal uses, including a horse vaccine against the West Nile virus. Advantages and disadvantages of nucleic acid vaccine. Immune response involves B cell and T cells. No live components, so no risk of vaccine triggering disease. Relatively easy to manufacture. Some RNA vaccines require ultra-cold storage. Never been licensed in human. Booster shots may be required. How do nucleic acid vaccines trigger immunity? In the case of DNA vaccines, a piece of DNA encoding the antigen is first inserted into a bacterial plasmid. This is a circular piece of DNA used by bacterium to store and share genes which will benefit its survivor, a bit like computer flash drive. Plasmids can replicate immediately of the main chromosomal DNA and provide a simple tool for transferring genes between the cells. Because of this, they are already widely spread used in the field of genetic engineering. DNA plasmids carrying the antigen are usually injected into the muscle, but a key challenge is getting them to across into people's cells. This is an essential step because the machinery which enables the antigen to be translated into protein is located inside the cells. Various technologies are being developed to aid this process, such as electroporation, where short pulses of electric current are used to create a temporary pause in patients' cell membrane, and gene gun, which uses helium to propel DNA into the skin cells and encapsulating the DNA in nanoparticles which are designed to fuse with the cell membrane. RNA vaccines encode the antigen of interest in messenger RNA mRNA or self-amplifying RNA saRNA, molecular templates used by cellular factories to produce proteins. Because of its transitory nature, there is a zero risk of it integrating with our own genetic material. The RNA can be injected by itself, encapsulated within the nanoparticles as Pfizer mRNA-based COVID-19 vaccine is, or driven into the cells using some of the same technique being developed for DNA virus. DNA vaccine. Once the DNA or RNA is inside the cell and it starts producing the antigen, these are then displayed on its surface, where they can be detected by the immune system and trigger a response. This response includes killer T cells which seek out and destroy infected cells, as well as antibody producing B cells and T cells which support antibody production. How easy are they to manufacture? Once a pathogen gene has been sequenced, it's relatively quick and easy to design a vaccine against any of its protein. For instance, Moderna RNA vaccines against COVID-19 enter clinical trials within 2 months of the SARS-CoV genome being sequenced. This speed would be particularly important in the face of new emerging epidemic, pandemic pathogen or pathogenic which are rapidly mutating. Both the DNA and RNA vaccines are relatively easy to produce, but the manufacturing process differs slightly between them. Once the DNA encoding the antigen has been chemically synthesized, it is inserted into a bacterial plasmid with the help of specific enzymes, a relatively straightforward procedure. Multiple copies of the plasmid are then produced within the giant vats of rapidly dividing bacteria before being isolated and purified. RNA vaccines are easier to synthesize because this can be done chemically from a template in the lab without the need for any bacteria or cell. In both cases, vaccines from different antigens could be manufactured within the same facilities, further reducing cost. This is not possible for most conventional vaccines.
That's all from me now guys. I hope from this video you'd gain more knowledge, and if you like this video please give me a thumbs up and please put a comment below this video what other video you'd like me to cover so that I know what you're looking for, and please do share and subscribe to my channel for my friend Justin.